Hi everyone, today we are going to talk about Chapter 19, which covers brain rhythms and sleep. In today's lecture, we're covering all things sleep. We're talking about circadian rhythms, we'll discuss EEGs and how they can help us measure brain waves. We're going to discuss sleep stages, why we need sleep. We're going to talk about REM sleep and dreaming, the neural mechanisms of sleep, sleep disorders, sleep hygiene, and how decreased sleep can actually increase our risk of several diseases. So, why is sleep research so important? It's amazing to think that we spend over 25 years of our lives sleeping, yet we know little about it. So, in all this time that we're sleeping, about a third of our life, our brain is working very hard to take care of our body. And we're going to talk about all the ways that the brain works hard while we're at sleep at night. Ultimately, the National Sleep Foundation and Sleep Research Society states that adults should be getting about 7 to 9 hours of sleep per night. Interestingly, newer research has actually demonstrated that women should be getting more sleep than men. Some studies have recommended that women should get about 8 to 10 hours of sleep, and women may even require an additional hour of sleep during menstruation. So ultimately, the amount of sleep that we need per night depends on multiple factors. How well and how much sleep you receive impacts everything else in your life. And not getting enough sleep actually increases your risk of several diseases. At the end of today's lecture, I'm going to talk about how chronic sleep restriction or chronic sleep loss essentially increases your risk of dementia and Alzheimer's disease. What is circadian rhythm? I'm sure you guys have heard of this before, but essentially, circadian rhythm is our daily cycles of physical and behavioral changes that roughly follow a 24-hour clock cycle. So, as you can see here in this figure, this is showing circadian rhythms of physiological functions. And there's fluctuation of our circadian rhythm throughout the day. Ultimately, this is naturally occurring and it's entrained by the environment. We have different cues that actually affect our circadian rhythm. One of these cues is known as a Zeitgeber. It's a fun word to say, Zeitgeber, but this is a cue in the regulation of the body's circadian rhythms. For example, this could be the light-dark cycle. So as mentioned previously, Zeitgebers are these external cues in the environment. So in this image, I wanted to point out that a Zeitgeber could simply be the light-dark cycle, this 24-hour cycle. When the sun rises in the morning, our retina is going to take in that light and communicate with the suprachiasmatic nucleus. This is our endogenous circadian rhythm and master clock regulator. So you have heard me talk about the suprachiasmatic nucleus before and how it does really function like the brain's master clock. The suprachiasmatic nucleus communicates with other areas of the brain like the pineal gland in order for us to synthesize melatonin. If you remember, we've talked about how we produce very low levels of melatonin throughout the day because this light cycle cues the SCN and the pineal gland to produce lower levels of melatonin. Whereas at night, this Zeitgeber or dark cue is taken in by the retina, and the SCN communicates with the pineal gland to produce higher levels of melatonin because it recognizes that it's time for us to go to sleep. Ultimately, we have other Zeitgebers that actually help us cue into our environment around us and to maintain our circadian rhythm. These Zeitgebers could include our sleep-wake cycle, physical activity, social time, and meals. I'll use the example of meals. We have breakfast, lunch, and dinner throughout the day. And when we consume a meal, it actually works as a Zeitgeber to help us maintain our endogenous circadian rhythm. Now, we can follow circadian rhythms without Zeitgebers, and some studies have actually demonstrated that if you remove cells from the suprachiasmatic nucleus and grow them in culture in a petri dish, that they can still maintain a circadian rhythm even without these Zeitgeber cues. I always utilize my cat Oliver as a perfect example of utilizing Zeitgebers. Mealtime is especially important to Oliver. He lives for breakfast and dinner. And we actually have an automated feeder that we use in our house. So my husband and I have the feeder set at 7 a.m. and 5 p.m. every day. And interestingly, Oliver knows when mealtime is approaching. He has this endogenous clock built in. And the Zeitgeber for him is mealtime. So this activates his master clock, his suprachiasmatic nucleus. Really, it's honestly hilarious. At around 4:45, Oliver will start to approach the food bowl and feeder and wait for it to release his food exactly at 5 p.m. every day. So this is a great example of an external cue or Zeitgeber helping him maintain his master clock. Light is so important for our mental health and our physiological well-being. You've probably heard of a psychiatric disorder called seasonal affective disorder, or SAD. Now, this disorder is more common during the winter months when we have shorter day length. When we have less access to sunlight, this can actually affect our circadian rhythm. Patients with SAD become more depressed in the winter due to a delay in circadian rhythms with respect to the sleep-wake cycle. And SAD patients usually respond to antidepressant effects like bright light exposure when it's scheduled in the morning. So this is a great therapeutic strategy to help decrease symptoms of SAD. And this also helps to restore the optimum alignment between circadian rhythms that are related to the sleep-wake cycle. So, for example, bright light therapy in the morning time could mimic the daylight that we experience naturally, which can activate the brain's master clock, the suprachiasmatic nucleus. Some symptoms of SAD include sleeping more than usual, increased appetite, and carbohydrate cravings. Now, this is interesting because some carbohydrates have higher levels of tryptophan. And so thus, if you intake carbohydrates, you could potentially increase tryptophan, which is the amino acid that's a precursor for serotonin and melatonin. Other side effects include weight gain, irritability, heaviness in the arms or legs, and social issues. So ultimately, bright light therapy could be an important way to restore circadian rhythms with respect to the sleep-wake cycle. And if you want to learn more about seasonal affective disorder, you can take a look at this link that I posted below. So as I previously mentioned, the suprachiasmatic nucleus is the brain's master clock. This is referred to as the SCN, and it's shown here in green. It sits right here, ventral to the hypothalamus, and dorsal to the optic chiasm and pituitary. So this is our brain's master clock. The SCN is a biological clock that produces circadian rhythms and consists of several different components. When we take in light, the retina receives that light and sends that information to the SCN. And the SCN then has an output pathway where it can send this information. Interestingly, in experiments, when the suprachiasmatic nucleus is stimulated electrically, circadian rhythms can actually be shifted in a predictable way. We also know that the brain's master clock is helping to regulate melatonin release from the pineal gland. And that's why it's so important to keep good sleep hygiene. We're going to talk about how too much screen time late at night can actually activate our brain's master clock and essentially inhibit the ability for us to produce melatonin late at night. The suprachiasmatic nucleus is absolutely essential for maintaining proper circadian rhythmicity. So studies have shown that if you remove both nuclei of the suprachiasmatic nucleus, this abolishes circadian rhythmicity of physical activity, sleeping and waking, and feeding and drinking. And so circadian rhythms can be abolished in monkeys that have lesions in both suprachiasmatic nuclei, and they're kept in the same constant light environment. Interestingly, in this image here, you can notice that persistent high frequency rhythms of both activity and temperature result from lesions in the suprachiasmatic nucleus. As you probably inferred, of course, jet lag and traveling affect our circadian rhythmicity. It's interesting that it's harder for our body to resynchronize when we travel from west to east, because during this time we actually lose time, and we're not used to this. So as you can see in this image, it takes a longer amount of time, a longer amount of days specifically to resynchronize when we're traveling, let's say, from the United States to Africa or Europe. And so thus, if you travel east over many different time zones, you're losing additional time. And this can make it harder to resynchronize back to your normal circadian rhythm. This is also why we may have a harder time adjusting to the spring forward during daylight savings time. Ultimately, this process is going to disrupt our body's natural circadian rhythm. Next, we're going to talk about how we can record brain waves through EEG measurements. We've previously talked about the EEG in class before. This is a measurement of electrical activity from the surface of the scalp. And in reality, this actually allows us to look at generalized activity in the cerebral cortex. If you remember, we talked about this device that's placed on the scalp that allows neuroscientists to actually record activity from the cortex. In my previous lectures, I've talked about how EEGs are primarily utilized to help diagnose different neurological conditions, especially seizures or epilepsy. If you remember, I've used an example before where we could look at a generalized seizure versus a specific seizure in a certain area of the brain. Ultimately, EEGs are also really helpful in sleep research. Researchers can utilize EEGs to actually study the stages of sleep and cognitive processes during wakefulness. And next, we're actually going to talk about synaptic currents that can occur in pyramidal cells. As I previously mentioned in other lectures, an EEG measures voltages generated by the currents that flow during synaptic excitation in the cerebral cortex. So the EEG device is laying on top of the scalp. And during this time, it's actually recording the activity of neurons below it. The electrical contribution of any single cortical neuron is very small, so the signal needs to penetrate several layers to actually reach the electrodes. Thus, it's going to take thousands of neurons that are activated together, firing together to generate an EEG signal that's big enough to be measured. And so the amplitude of EEG signals strongly depends on how synchronous neurons are. So if we have increased synchronous activity of cells, meaning cells are firing action potentials together, this is going to increase the surface signal recognized by the EEG electrode. So when a group of cells is excited simultaneously, this is going to increase the ability for us to record these brain waves. So now let's talk about the synaptic currents in pyramidal cells. So the active synapse is on the upper part of the dendrite shown here. And when the afferent axon fires, the presynaptic terminal is going to release glutamate. We know that glutamate induces excitation in neurons. And this is going to open up cation channels and lead to depolarization. Positive current flows into the dendrite, leaving a slightly negative extracellular fluid. Current spreads down the dendrite and escapes from its deeper parts, leaving the fluid slightly positive at those sites. And ultimately, the EEG electrode is going to measure this pattern through this thick layer of tissues. If we have thousands of neurons that are firing together simultaneously, this is going to increase the ability for us to measure that signal. And so, essentially, a key role of measuring neuron activity is this release of glutamate that's going to induce excitation and depolarization in a large group of cells. Here's a great example of this. Now let's look at a population of pyramidal cells that are located underneath this EEG electrode. So each neuron receives many different synaptic inputs, as shown right here. Now, if the input fires at irregular intervals, as shown in the top right here, this means that the pyramidal cells are not firing in a synchronous manner. This is going to only create a small amplitude of activity. However, as shown below in the synchronized image, if thousands of neurons are firing simultaneously and have many action potentials that are occurring at once, this synchronized activity is going to result in a much larger response that can be recorded by the EEG. So we've talked a lot about how we can measure these electrical rhythms in the cerebral cortex with an EEG, but how are these rhythms generated and what functions do they perform? Well, a large set of neurons, which can include thousands of neurons, can produce synchronized oscillations in one of two fundamental ways. They can take their cues from a central clock, known as a pacemaker, or they can share and distribute the timing of their function among themselves by mutually inducing excitement or inhibition. So ultimately, there are two mechanisms of synchronous rhythms, either A, the pacemaker, which I just talked about, which is like a central clock, or these rhythms can arise from a collective behavior of neurons. So for example, in image A here, this is demonstrating synchronous firing. So for example, when we're asleep, we have increased synchronous firing at a lower frequency and a high amplitude. Whereas when we're awake, we may experience neurons in the cerebral cortex that are asynchronous, meaning they are firing at different times and they have higher frequency and lower amplitude. Now let's talk about the stages of sleep. So one of my best friends and my previous lab partner is a sleep researcher. And a lot of my material from this lecture I actually received from her, but she has told me all about these different stages of sleep. So essentially in stage 1 of sleep, this is when we experience theta rhythms. And this is essentially occurring during that transition from waking to sleep. So there's a drop in high frequency firing. Stage 2 is when we're still experiencing the theta rhythms. This is known as light sleep stage, and it's essentially like the first true stage of sleep. So at this time, our body temperature actually decreases and we can experience non-REM dreams. They may not be as vivid as our REM dreams, but they can occur during this stage. Now, stages 3 and 4, as shown here in this light purple color, are when we experience these delta rhythms. This is known as our recovery sleep or our deep sleep. When we experience delta rhythms, we have decreased rate of respiration and our heart rate and our respiration slow, our muscles relax. So this is our really good recovery sleep. And as you can see in this graph over here, which I forgot to introduce earlier, on the y-axis here, this is showing us our different sleep stages, and on the x-axis, this is showing us sleep from 11 p.m. to 6 a.m. And as you can see, we have these 90-minute cycles in which we can experience these different stages of sleep. And in purple here, we can see where we experience that good, deep recovery sleep. Finally, REM sleep, which REM stands for rapid eye movement, is when we experience these beta rhythms. And essentially during REM sleep, our heart rate and our respiration rate increase. And so during this time, we actually have a drop in muscle tone as pathways to the spinal motor neurons are actually inhibited. In adults, REM accounts for about 25% of our total sleep time, and during this time is when we are likely experiencing these vivid emotional dreams. So if you wake up in the morning and you can remember a crazy, vivid dream that almost felt real to you, that was likely a dream that occurred during your REM sleep. So why do we need sleep? Obviously, we know sleep is super important for different mechanisms like synaptic pruning, but ultimately, all organisms show at least a primitive form of sleep, and different mammals require different amounts of sleep. For example, giraffes usually average less than 5 hours of sleep per night, while some types of bats can average 15 to 20 hours of sleep per day. Even animals without brains like jellyfish show some primitive form of sleep. And other mammals like cats, of course, my cat probably gets about 20 hours a day. He's sleeping all the time. One of my favorite mammals to talk about in the context of sleep is the Atlantic bottlenose dolphin. This mammal conducts something called unihemispheric sleep, and it can actually sleep with only one hemisphere of the brain active. So essentially, the Atlantic bottlenose can sleep with only the right half of their brain active, while the other left half remains alert. And this is very adaptive and advantageous because it increases their survival, right? It's good that one hemisphere is alert while the other is at least getting rest. And in this picture, this is showing the left and right hemisphere of the Atlantic bottlenose dolphin. And in image A here, we can see that there is different EEG architecture. So in A, this is demonstrating alert wakefulness on both sides of the brain. And then in image B, this is showing us intermediate sleep on the right side while the left side is still remaining active. In image C, this is demonstrating deep sleep on the right side of the brain while the left side is staying alert in case predators are coming along. And then in image D here, this is when the pattern is shifting to let the other side of the brain sleep. So now the left side will be receiving sleep. Interestingly, there's another type of dolphin called the blind Indus dolphin that can't actually go to sleep for long periods of time. So it gets something called microsleeps. It actually only sleeps for 5 to 6 seconds at a time. So ultimately, mammals have evolved to develop different types of sleep patterns. And it's really interesting to look at how different species can sleep. Now let's talk a little bit about REM sleep and dreaming. And of course, I had to include a picture from Breakfast at Tiffany's because Audrey Hepburn's eye mask in this movie is absolutely iconic. Studies have actually shown that if you have less light in your room, it can help you to sleep, and it's also been shown to help increase melatonin production. So sleeping with an eye mask is actually a great sleep hygiene tip, which we'll talk about more at the end of today's lecture. Back to REM. REM stands for rapid eye movement, and during REM we have a loss of muscle tone, and we typically experience dreams that are more vivid or emotional. If you remember a very vivid dream when you wake up in the morning, this dream likely occurred during REM. REM is also really important for building new synapses, so thus it's playing an important role in learning and memory consolidation. You can think of REM as this time where we're recording and editing synapses built throughout the day. Also, neural protein synthesis actually peaks during REM sleep. Other research has found that non-REM sleep is great for pruning away unnecessary synapses, and we know how important that is for neural health. So as I previously mentioned, synaptic pruning largely occurs during non-REM sleep. However, I want to keep you guys up to date with the newest scientific literature. And because sleep research and the effects of sleep on the brain are still so under-researched, it's really important to keep looking at new journal articles that are being published. And so I want to talk about a more recent study that was published. I provided a link on Canvas for you guys if you want to take a look at this paper. This was a study that was conducted in mice, and they found that REM sleep prunes newly formed postsynaptic dendritic spines of pyramidal neurons in the mouse motor cortex. And so this pruning was largely occurring during development and motor learning. Now, in this study, synaptic pruning in REM helped to balance the number of new dendritic spines that formed over time. So this was really cool, as most research has usually demonstrated that REM sleep is responsible for strengthening synapses rather than synaptic pruning. But in this study, they also found that REM sleep strengthened and maintained the newly formed dendritic spines that were critical for neuronal circuit development and behavioral improvement after learning. So ultimately, because this is such a new field and we still don't know a lot about the effects of sleep on the brain, it's important to look at new updated research. Why do we dream? Well, I don't have a great answer for this. Neuroscientists are still trying to investigate the biological importance of dreaming, but there's a lot of hypotheses as to why we dream. Perhaps dreaming plays an important role in memory consolidation. We know that synapses are strengthened during sleep, especially during REM sleep. So maybe this is a way to enhance the consolidation of newly created memories from the day. Perhaps it's a way for us to resolve anxiety and issues that we weren't able to resolve during the day. Perhaps it helps us with problem solving or coping with stress. We've all had, probably all of us have experienced these stress dreams that sometimes are reoccurring. And so I do think that sleep and dreaming is a great way for us to try to resolve unresolved issues like stress and anxiety. But ultimately, the actual biological importance of dreaming is still being investigated. Now I'm briefly just going to talk about sleep hygiene. Now, sleep hygiene is just as important as your daily hygiene, so I encourage you to utilize these sleep hygiene tips in your own life. So ultimately, I've talked about how staying up late at night with screens can be really detrimental to our health, mainly because if we have increased light exposure at night, that can affect the suprachiasmatic nucleus and its communication with the pineal gland. If we look at screens too late at night, we may suppress melatonin production, and we know melatonin is super important for allowing us to get a good night's sleep. So here are some good sleep hygiene tips. First, sleep researchers recommend that you try to go to bed at the same time every night and wake up at the same time every morning. This even is important during the weekends. I know this can sometimes be really difficult, but again, this is really important. Having a consistent nightly routine is really important because it helps your brain to wind down. I know there's some great applications on the iPhone. We even have the sleep reminder on the iPhone that can tell you when it's time to start winding down before bed. So that's a great reminder if you can't remember to create a nightly routine on your own. It's also important to note that you should never take an afternoon nap longer than 2 hours. This is absolutely going to disrupt your sleep at night. Limit caffeine intake. This one is a given. Make sure you don't have caffeine too late in the afternoon. Avoid exercising within an hour before going to bed. This is super important because when we exercise, we release endorphins and other important chemicals that could keep us up. Avoid using tobacco, alcohol, or caffeine within 4 hours of going to bed. Avoid using technology about 30 to 60 minutes before bed. This is so crucial. When we have access to blue light, this is going to suppress melatonin release. Remember, melatonin is going to help us with that onset of sleep. And unfortunately, in today's world, we all do this. We're looking at screens late at night, but I really encourage you to try to put your phone or laptop away 30 to 60 minutes before bed so you can allow your pineal gland to release the proper amount of melatonin. Make sure your room is cool, dark, and quiet. A cold room will usually help with sleep. Avoid going to bed feeling anxious or stressed or angry. So I know this can be really difficult to do. So you could try to utilize meditation exercises. You could try to pray before bed, anything that can help you relax and decrease your anxiety. Avoid using your bed for anything other than sleep and sex. Unfortunately, if you're working in bed, your brain is going to associate your bed with your awake day activities, and this can increase arousal and wakefulness rather than sleep. Try to avoid checking emails or texts during your sleep time. If you look at your work email when you're trying to sleep, that can activate anxiety. And turn your phone on silent so that it doesn't wake you up. So this is super important. Also, make sure that you exercise during the day. If you exercise during the day, this is going to make you feel more tired at night and might make it easier to fall asleep. Now, to end today's lecture, I wanted to talk about how sleep loss can actually potentially increase your risk of disease. And specifically, chronic sleep loss has actually been associated with an increased risk of dementia and Alzheimer's disease. Now, in class, I've briefly mentioned that in my laboratory at Texas Christian University, my lab mates and I studied the effects of chronic sleep restriction on Alzheimer's disease pathologies and cognition in wild-type mice. Now, remember, experiments utilizing wild-type mice are different than those that utilize transgenic mouse models of Alzheimer's disease. Wild-type mice don't have trans genes inserted into their genome like a transgenic mouse model of AD. If you remember, I talked about how trans genes are actually inserted into the mouse genome in order to induce Alzheimer's disease pathologies in transgenic mouse models of AD. Our laboratory specifically focused on how environmental risk factors like chronic sleep loss could actually induce the development of Alzheimer's disease pathologies in a wild-type mouse, which is kind of like a healthy human model, right? This is not a mouse that's predisposed to Alzheimer's disease. And ultimately, we found some really cool things. So you can check out this paper. We published it in Physiology and Behavior. Again, this paper is actually available on Canvas if you want to read it. But I'll talk briefly about the results of our study on the next slide. Our laboratory investigated the effects of chronic sleep loss on Alzheimer's disease biomarkers in wild-type mice. And the biomarker we chose to focus on was amyloid beta, as shown here on the y-axis. If you remember, we've talked about amyloid beta in class before. This is a peptide that in a non-pathogenic state is just produced as a monomer, and this is a normally occurring peptide in the brain. However, in a pathogenic state in the Alzheimer's disease brain, amyloid beta has the potential to aggregate together, and this can form into extracellular amyloid beta plaques. These extracellular plaques actually disrupt neuronal communication, inhibiting synapses, and ultimately can lead to neuron death. This is a key hallmark pathology of Alzheimer's disease. So ultimately, this was the biomarker we chose to focus on. Now, I will very briefly mention the methodology of this experiment, because a lot of people ask me, how do you induce chronic sleep loss in mice? We utilized this method called the modified platform method. And essentially, in this method, there's a cage with platforms, and underneath the platforms, we fill a tub with water. Now, based on today's lecture, we know that when you go into REM, you lose muscle tone. So if mice on the platforms went into REM, they would fall off the platform into the water. Thus, this paradigm prevents mice from going into REM sleep as they would lose muscle tone and fall in the water. So this is a widely used method for chronic sleep restriction in mice. We also had two control groups in which mice could sleep whenever they wanted. So ultimately, mice were kept in chronic sleep restriction or their control cages for a 6-week period of time, but they were only placed in the chronic sleep restriction cages for 10 hours of the day. We didn't want to inhibit their sleep throughout the entire 24-hour day, as this would be looking at severe sleep deprivation. So each day, the mice were placed in this cage for 10 hours, either the sleep restriction cage or the control cages, and then they were put back in their home cage at night. So they still were able to sleep. This was just looking again at the effects of chronic sleep loss to try to mimic what a human may experience if they lose a few hours of sleep at night. Ultimately, we found that after 6 weeks of chronic sleep restriction, that was 10 hours a day over the course of 6 weeks, mice that were chronically sleep restricted had higher levels of amyloid beta protein compared to mice that were in the control cages. Ultimately, this is one of the first studies to actually demonstrate that chronic sleep restriction increases amyloid beta in wild type mice, and thus chronic sleep loss could be a risk factor for sporadic Alzheimer's disease. This is a subtype of Alzheimer's disease that's more prevalent, and the etiology of it is still largely unknown. So, if you remember anything from today's lecture, I hope that you can take away a few key things. Make sure you look at your sleep hygiene tips and make sure that you're taking care of yourself to get enough sleep. And remember that sleep loss could be a risk factor for different diseases like Alzheimer's disease. So make sure you're taking care of yourself and getting enough rest.